As I mentioned, the nucleus determines both the structure of the cell and the functions it can perform, and it does this by controlling which proteins are synthesized, under which circumstances, and in what amounts. So the next sections explore the process of protein synthesis, which can be divided into two stages, transcription and translation. Transcription involves the production of RNA from DNA. So essentially we're making a DNA copy, which is called messenger RNA, and that messenger RNA is used for protein synthesis from the nucleus out to the cytosol. Translation, then, is the construction of protein from that RNA. So the mRNA leaves the nucleus and binds to a ribosome, and this could be either in the cytoplasm or on the RER, the rough endoplasmic reticulum. And transfer RNA, a different type of RNA, tRNA, delivers amino acids to those ribosomes. Each tRNA has a complement to the codon called an anticodon, and each amino acid has one or more codons. So sometimes a picture is worth 1,000 words. This picture is showing 3 steps of transcription. And first of all, RNA polymerase binds to what we call a promoter sequence, and a promoter gene. So it's a very particular sequence of the DNA, and it unzips the DNA by breaking hydrogen bonds between the double helix. So the promoter gene is a section of DNA that acts kind of like a light switch to turn on a gene or to turn it off. So to use that gene or to not use it to make proteins. So then the second step, complementary RNA codon triplets are formed, and this is using uracil instead of thiamine. If you notice, remember that complementary base pairing I told you about? Guanine tells RNA to make cytosines. Guanine, cytosine. Cytosine tells it to make RNA guanine. And then adenine, the complement will be uracil. So that's how we know this is an RNA molecule instead of a DNA molecule. And then step 3, once that RNA polymerase hits a stop codon, it will be released from the DNA, and the DNA will re-zip itself shut so that our mRNA can be released to be used to make proteins. So that's the 3 steps of transcription in a nutshell. And then we use that RNA for translation. So that mRNA is going to have to leave the nucleus, going to go out through one of those nuclear pores, leaving the nuclear envelope behind, and it's going to be looking for a small ribosomal subunit, which is what we have pictured, this big blue bubble right here. So the mRNA is going to combine with a small ribosomal subunit, and the first tRNA will bind with the start codon on the mRNA, and the start codon is always AUG. Okay, so the complement of A is uracil. The complement of A here is another uracil, right? That's complementary base pairing. Guanine goes with cytosine. And so this is a tRNA that has an anticodon that is complementary to this start codon. So the tRNA binds, and this is your first amino acid. Then the next step is this large, the other half, the large subunit of the ribosome comes together and wraps around the mRNA, the tRNA, and the small subunit. Okay, and so now you're ready to start cranking it out, because the second tRNA will then be able to enter, the anticodon will complementary base pair with the next triplet, the next codon. Okay. And what happens next is almost magical, or at least I think so anyways. You have these 2 amino acids here, amino acid 1 and amino acid 2, and after the second tRNA is bound in this active site, you form a peptide bond right here. And the ribosome will slide down the mRNA, almost like a typewriter slides down. Okay, so it's going to slide down one more codon, another triplet of the mRNA, And when it slides down, what happens is this first tRNA gets kicked out. The second tRNA slides into this first spot. And so now we have this open active site. And a third tRNA with a anticodon that's complementary to the next codon can then bind here and bring the third amino acid. And then we're going to form a peptide bond here, and this continues all the way down this mRNA strand. And they have a very, very brief mRNA strand here just to show you how you can then form this polypeptide chain. Once you reach the stop codon at the very end, the stop codon is UAA, the polypeptide chain is released, the small subunit breaks apart from the large subunit, and the mRNA is then degraded because you're done with it. And you've created your protein, your polypeptide. And so in those two slides, that is the process of translation then. And so these are some examples of the triplet code. So if your DNA reads AAA, your mRNA or your complementary binding would be UUU. Your tRNA's anticodon, the part that binds to the mRNA, is always going to match the DNA. Okay, at least two of the three will always match. And so this anticodon will determine which amino acid is attached to that tRNA. And so you have a DNA triplet, which codes mRNA, which binds to an anticodon on the tRNA, which brings different amino acids depending on the code. This is how you can use the DNA triplet code to determine a protein sequence. And so that brings us to another checkpoint, and you can pause the recording and see if you could describe to me how the nucleus controls the cell's activities, and your description should contain transcription and translation descriptions. Moving on to section 3-8, we're going to be now describing the stages of the cell's life cycle. And you might have noticed over the course of your life that your body has this amazing quality of regeneration. And so you get a cut on your hand, right? And eventually new skin forms and closes that wound. No matter how many times you cut your hair or your fingernails, they keep on regrowing. So this is because your somatic cells or your body cells have the capacity to perform cell division. And in terms of cell division, there's some important ideas to differentiate. So mitosis is the cell division that your body cells, like your hair or your nails or your skin, that those cells can go through. That's mitosis. However, to generate gametes like an egg cell or a sperm cell, these gametes that have half the number of chromosomes as the rest of your body cells, your sex cells must go through a very specialized form of cell division, which is called meiosis. Okay, and this process of meiosis will be discussed more in chapter 19, so stay tuned. We'll come back to that. And then last, there's one more term for me to present to you when talking about cell division, and that's apoptosis. Apoptosis is this fancy term for programmed cell death. Now, only stem cells can undergo mitosis, and the resulting regular run-of-the-mill tissue cells have this predetermined lifespan that ends when the cell triggers apoptosis. So we'll start our discussion of the cell's life cycle with interphase, because interphase, which is pictured in blue here, interphase is it's pretty much the phase where the cell spends most of its functional time. And I was making the quotation marks with my fingers when I said functional time. So interphase is the regular part of the cell. Interphase itself can be divided into three distinct phases, the G1, S, and G2 phases. So if you're one of the stem cells, let's say you're a skin stem cell and you're an interphase, you're hanging out in G1 phase, but you want to go through mitosis and make a new cell to replace a skin cell that has recently been sloughed off. So the first step is the G1 phase, and this is when the cell will duplicate all of its organelles and start massing cytosol, increasing its cytosol. The S phase of interphase then is when the cell will go ahead and replicate its DNA, make a copy of its DNA inside of the nucleus. Last, G2 phase of interphase is when the centrolaires are replicated and proteins are synthesized. Okay, so the cell is really gearing up for that mitosis, that next step. So before we get into mitosis, let's take a closer look at the S phase where DNA replication occurs. And in replication, the DNA strands unwind, and DNA polymerase, this special enzyme pictured here and here and here as big blue balls, the DNA strands unwind, and these DNA polymerase enzymes begin attaching complementary DNA nucleotides along each strand. And so the end result is that two identical copies of the original DNA molecule are produced, one with an old strand and one with a new strand. So this is called semi-conservative DNA replication. And this all happens in S phase, even before the cell enters mitosis. So let's take a look at mitosis. Here's interphase, and we're at the end of interphase. We've gone through G1, S, and G2 phases, and we're about to enter mitosis. Okay, so mitosis, depending on which textbook you read, is divided up into a number of stages or phases. Early prophase and late prophase. Older textbooks, it just calls it prophase. But prophase is when the DNA starts to coil really tightly enough so that it can actually be seen as a chromosome. And you can see this with a light microscope. You can see chromosomes inside of the nucleus. And each of the two copies of DNA is actually called a chromatid. Okay, each of the two copies is called a chromatid. And you can see them labeled here as sister chromatids. But this is a chromosome. And they're each connected to each other at a point called a centromere. So this is this little button in the middle, a centromere that buttons those two sister chromatids together. The nucleus starts to disappear, okay, and the two pairs of the centroles, which are pretty much very, very difficult to see, if at all, unless you're in mitosis, they appear and they start to move to opposite ends of the pole, the top and the bottom. And you have these long finger-like projections called spindles that start to appear. Okay, so this is prophase. And at the end of prophase, you then enter into metaphase. And technically, metaphase is this really, really brief blip of time where the chromatids are all lined up in the middle of the cell, perfectly lined up on what we call a metaphase plate. Okay, so they're parallel to the plate. And then this is so brief, and the next section, the next phase, is anaphase. And this is where our sister chromatids are pulled apart. All right, so your chromosomes, it looks like they're split in half. But really what's happening is you're just pulling apart the sister chromatids. And so these daughter chromosomes are pulled apart, and they move towards the centromeres. They're moved towards either end of the cell in anaphase. And this blends right into the next stage, which is telophase. In telophase, your nuclear membrane starts to reform around your daughter chromosomes. Okay, the DNA starts to uncoil. It no longer is looking like that typical chromosome structure. And the cells are preparing to enter into interphase again. So they're starting to normalize one more time, and these spindle apparatus are going to start to slowly break down. Okay. After the cell's nucleus and its contents are appropriately divided, that's the last stage in the cell's mitosis phase, because then it enters into this process called cytokinesis, where the cell actually splits in half. So cytokinesis is that division of the cytoplasm. It forms two separate daughter cells. It usually begins in late anaphase, continues through telophase, and it's completed after that nuclear membrane has reformed, and around each daughter nucleus. And so then each of these daughter cells, which, by the way, are genetically identical, then enter into interphase. Now, if this is the stem cell, it'll probably go through mitosis again at some point, and this one, if it's just a regular cell, will move out towards the surface of your cell and then be sloughed off or go through the process of apoptosis. So can you define mitosis and list its 4 stages? Why don't you pause the recording and see if you can do that just from memory quickly. If not, go ahead and review and then move on to the last section of this recording, which will be cell differentiation. So cell differentiation is just this little blip in your textbook at the end of this chapter that describes how all of your somatic cells in your body have the exact same chromosomes and genes. It's all there. Whether you're a skin cell or a liver cell or a nerve cell, it doesn't matter. They all have the same exact chromosomes. They all have the potential to have all of those same genes, okay? And that's why, like on CSI or some crime show, it doesn't matter if they find a hair or if they find skin cells or whatever. They can get the person's entire genetic makeup from any body cell, okay? But yet we have this at the beginning of this chapter, I told you that we have a wide variety of cell types, right? So how is that possible if they all have the same chromosomes, the same genes? Well, it's called differentiation. And remember I told you those promoter genes are like little light switches that turn on or turn off specific genes? Well, those promoters are either flipped on or flipped off, and they leave the cell with only so many genes that are functional. And so it kind of determines what the cell will look like depending on which light switches are flipped. Okay, they all have the genes, they're all present. It's just which ones are on, which ones are active. Okay, and so the cells can then become very, very specialized. And the collection of these cells all together, working together, is called a tissue. Okay, and we have a number of different tissues in our body, and we'll talk about those in the upcoming chapters. But I just want to bring up this picture one last time to show you all of these cells. If you cracked open their nucleus, inside, they have the exact same chromosomes, the exact same genes. It's just which ones are turned on, which ones are expressed to give you these completely different structures, these completely different functional cells. Okay? And again, we'll talk about that more in the upcoming chapters. So this is the end of chapter 3.